We are going to track the human machine through a specific theoretical model, 30 consecutive days in total soundproof isolation. At day 0, the subject enters the chamber with healthy baseline vitals, normal blood pressure, stable endocrine function, and standard neural activity. To understand what happens next, we have to look at the foundational biological classification of our species. Humans are obligate gregarious animals. An obligate gregarious species is an organism that requires association with its own kind for its physiological processes to function normally. Our biology relies on the continuous presence of others to regulate its internal systems. Sensory deprivation is frequently assumed to provide neurological rest. The empirical reality is the exact opposite. Entering the absolute void of a soundproof room induces immediate biological shock. Removing all social connection severs a critical evolutionary life support system. The true impact of this environment occurs entirely under the skin, driven by how the internal hardware processes the silence. When confronted with a sudden, absolute absence of social and sensory input, the brain runs an immediate threat assessment. Ancient neural structures evolved in environments where isolation meant certain death from predation or starvation. Consequently, the brain interprets total isolation as an immediate lethal threat to physical survival. Over the next 30 days, we will track the biological telemetry of this threat response as it spreads through the body. The sequence of collapse occurs in 3 distinct phases: a runaway endocrine stress cascade, a transcriptomic shift in the immune system, and finally, the physical degradation of brain tissue. The ensuing damage is a measurable mechanical failure of the body's architecture. Within the first 72 hours, the speed of the body's reaction becomes violently apparent. The primary responder to the threat of isolation is the hypothalamic pituitary adrenal, or HPA axis. The HPA axis is a complex set of direct influences and feedback interactions among 3 endocrine glands that controls our reaction to stress. Under normal conditions, minor everyday social interactions provide subtle biochemical feedback. This feedback acts as a biological brake, telling the HPA axis that the environment is secure and allowing stress hormone production to ramp down. Looking at this diagram of the HPA axis, we can observe the mechanical failure. The designated social buffer input line is severed. Without that input, the system loses its braking mechanism and locks into an infinite feedback loop. The immediate output of this broken loop is shown here on the graph. Cortisol levels violently spike and plateau at maximum capacity. The body is physically incapable of bridging the gap back down to the safe zone. It requires external social cues to signal safety and lower the cortisol output. By the 3rd day, the subject is permanently locked in a chemically unsustainable state of hyperarousal. Reaching day 3 marks the crossing of a critical physiological threshold. The brain's neural networks are now experiencing severe sensory starvation. To prevent its circuits from going dormant, the brain executes a drastic survival mechanism. It manufactures its own stimuli to maintain baseline neural firing patterns. Minor internal sounds, like a heartbeat or a breath, become deafeningly loud. Vague peripheral shadows are actively perceived as sentient beings moving through the room. These hallucinations indicate the social brain is starving. It is desperately cannibalizing its own resources to keep its sensory processing circuits alive. Advancing into days 4 through 14, the acute stress begins to cause chronic systemic desynchronization. The human biological clock is anchored by Zeitgebers. Zeitgebers are environmental and social cues that synchronize the body's internal rhythms to a specific schedule. The rhythm of eating a shared meal or engaging in a daily conversation provides the precise timing signals the brain needs to regulate waking and resting hours. This dial represents the body's internal clock. When those daily social anchors are stripped away, the neat sleep-wake blocks fracture, and the circadian rhythm begins free running. A free-running rhythm operates entirely independently of external time cues. This erratic sine wave shows the physical output. The subject might stay rigidly awake for 30 consecutive hours and then sleep deeply for 12. This temporal chaos triggers a secondary hormonal crash. The production of melatonin and growth hormone is severely suppressed. Without social anchors to regulate time, cellular repair halts, leading to rapid cellular aging and deep physical exhaustion. Simultaneously, the chronic high cortisol established in the first week begins to systematically dismantle the subject's metabolic health. We can observe this destruction by looking down at the microscopic level, specifically at the body's white blood cells. Deep within the nucleus of these immune cells, the isolation triggers a transcriptomic shift. A transcriptomic shift is a large-scale alteration in how a cell translates its DNA instructions into active proteins. This cross-section shows the immune cell's actual gene expression altering in direct response to the void. The internal DNA strands swap specific coding blocks, abandoning a protective chemical profile in favor of a highly inflammatory one. This newly altered genetic profile creates a severe physical vulnerability. As shown here, viral threats can easily bypass the cell's weakened defenses, prompting the release of cascading inflammatory markers. Rampant systemic inflammation takes hold in the tissues and organs throughout the body. Chronic loneliness quite literally reprograms the subject's DNA to dismantle their own immune defense system. The model now enters its final and most destructive phase, days 15 to 30. The persistent lack of external input results in actual, measurable physical mass loss inside the brain. This mass loss is concentrated in the hippocampus. The hippocampus is a complex brain structure embedded deep in the temporal lobe that regulates memory encoding and spatial navigation. The brain operates on a strict energy economy. It ruthlessly cuts off resources to pathways that are no longer stimulated by dynamic human interaction. This process is called neural pruning. Neural pruning is a biological mechanism where the brain actively dissolves unused synaptic connections to conserve energy. The macroscopic, lived experience of this shrinkage is profound. The subject experiences dense brain fog, active memory erasure, and a complete loss of time. Starved of human contact, the social brain physically eats itself to survive the silence. The complete 30-day biological telemetry paints a highly specific picture. The subject suffers from chronic elevated cortisol, a violently desynchronized circadian rhythm, pro-inflammatory leukocytes, and a physically shrunken hippocampus. Solitary confinement tests the structural integrity of the human machine. The resulting data points to a cascading mechanical breakdown of vital systems. Human biology cannot sustain its own physiological operations without the continuous regulatory tether of other humans. Pushed to its absolute limit, total solitude forces the human body to dismantle itself, from the expression of its DNA to the physical architecture of the brain.
Thanks for watching Body Science School. Please like, share, and subscribe the channel for more.